Hello students, welcome to EPG Pathshala. I am Dr. M.N. Gupta from IIT Delhi. Today we will talk on a module wherein we will continue to talk on hypersensitivity of the other kinds from paper Immunology. Type 4 hypersensitivity is also called delayed type as results are seen from 48 hours to 28 days. In the three variants of the type 4, the delay is of different time periods. We will also look at the type 5 hypersensitivity, which was introduced as a separate class after Coombs and Keller classification. This hypersensitivity is involved in some autoimmune diseases. So the objectives of this module are to understand the three types of class 4 hypersensitivity reactions, which are called contact dermatitis, tuberculin reaction, and the granulomatous hypersensitivity reactions. The second objective is to explain how mechanistically all the three types follow the same route, and to learn how certain diseases caused by infectious agents involve this type 4 hypersensitivity. We'll also understand type 5 hypersensitivity reactions which occur because of chronic stimulation. So the concepts involved are the delayed hypersensitivity, which consists of contact dermatitis, tuberculin reaction, and granulomatous reactions, and we will also cover the type 5 hypersensitivity. Contact with some substances can cause contact dermatitis. Injection of some antigens derived from infectious agents can be used as a test for hypersensitivity towards that microbe. When some microbes persist, it causes formation of granulomas. All are examples of delayed hypersensitivity. On the other hand, a variant of type 2 hypersensitivity, when antigen is in fact an antibody against a host antigen, causes chronic stimulation. This is classified as a type 5 hypersensitivity by some workers. In Coombs and Gale classification, all hypersensitivity reactions which take place more than 12 hours to develop and are dependent upon cell-mediated immunity are classified as type 4 or delayed type hypersensitivity. Much after this classification, late phase IgE mediated reactions were discovered. These are at their maximum in the time period of 12 to 24 hours after the exposure to allergen. As we have discussed, these also involve Th2 cells, so the definition of type 4 sensitivity is not adequate. Type 4 sensitivity cannot be transferred by serum but by T cells. T cells involved are sensitized T cells, that is, they had encountered the specific antigen, which in this context is called allergen, before and this time recruits other cells to the site of the reaction. Contact dermatitis appears within 72 hours of exposure to allergen. The exposure is through skin, which comes into contact with allergen. In Europe, the most common allergens are nickel, chromate, and chemicals in rubber. In USA, poison ivy and poison oak are reported to be responsible for maximum reported cases of contact dermatitis. In poison ivy, the responsible compound is urushiol, which is an oily mixture of catechol derivatives with long hydrocarbon side chains. Urushiol is secreted by the leaves of the poison ivy vine and related plants. Catechol derivatives are easily oxidized to reactive orthoquinones, which can react with cell surface molecules to generate allergens. The skin pigments melanin and melanoprotein in fact arises from similar reaction. Various metals like nickel and chromium present in jewelry, hooks and zipper of garments chelate with skin proteins to form allergens. Allergic dermatitis sometimes is observed on pathologist fingers because of constant exposure to formaldehyde, another reactive molecule. Other common chemicals are picric acid, aniline dyes, organophosphates, plant resins and oils. Unless the allergen is liquid, normally hairy parts of the skin are affected. 2,4-dichloronitrobenzene, abbreviated as DNCB, and similar compounds have been used to induce contact dermatitis in experimental animals. Most of these compounds which cause contact hypersensitivity act as haptens, react with skin cell surface molecules to form haptane carrier conjugates to become immunogenic. Contact hypersensitivity is distinguished from other type 4 reactions in being an epidermal phenomenon. The Langerhans cells, which is the antigen presenting cells in the skin, are the first step in the chain which is involved in this hypersensitivity. Langerhans cells originate from the family of follicular dendritic cells and are derived from bone marrow. These are about 3% of total epidermis cells. Within few hours, that is the 4 hours of using DNCB in mice, Langerhans cells appear in the draining lymph nodes. It is in lymph nodes that these cells act as antigen presenting cells and present antigen to lymphocytes. The first phase is sensitization phase. This consists of uptake, processing and presentation of the allergen by local antigen processing cells, antigen presentation cells. Sensitization takes place after 10 to 14 days in humans. Exposure leads to absorption. The haptens react with the skin protein and epidermal Langerhans cells internalize the antigen. The haptane containing peptides produced by antigen presenting cells are now ready to be presented. Antigen presenting cells, the Langerhans cells in this case, migrate to paracortical areas of regional lymph nodes and present the allergen to CD4 positive lymphocytes. These produce a population of memory T cells. These processes are shown in the slide here. In humans, both dose response as well as concentration that is the DNCB per unit area response correlations have been observed. There is a plateau beyond a certain level. DNCB amount per unit area determines the response. During the second and subsequent exposures, primed, that is the memory Th1 cells in lymph nodes appear at the skin. Triggered T cells secrete cytokines such as interferon gamma, which stimulate keratinocytes to secrete further cytokines and chemokines. Langerhans cells present the processed antigen in the form of heptane peptides to the memory CD4 plus T cells. CD3 receptor of the T cells is involved and T cells release cytokines which include IL-2, IL-3, interferon gamma and GM-CSF. The released interleukin-2 binds to the receptors on T cells and induce their proliferation. Interferon gamma and TNF induce epidermis keratinocytes to express intercellular adhesion molecule including ICAM1 in about 24 to 48 hours of the exposure to the allergen. At 48 hours, keratinocytes express HLA-DR under the influence of interferon gamma. Lymphocytes and macrophages have integrin LFA1 on their surface which recognize ICAM1. The result is that lymphocytes and macrophages come to the site in the skin. Activated keratinocytes also release interleukin-1, interleukin-6 and GM-CSF, promoting activation and proliferation of T cells. GM-CSF also activates Langerhans cells similarly. Endothelial cells in the dermis also express adhesion cells and can get involved in accumulation of lymphocytes to the site. The reaction begins to fade after 48 to 72 hours. This slide shows the cytokines, prostaglandins and cellular interactions in this process of contact hypersensitivity. At this point, when the reaction is beginning to fade, macrophages and keratinocytes produce prostaglandin E, which inhibits interleukin-1 and interleukin-2 production. T cells bind to keratinocytes. The heptane peptide is degraded by the cellular mechanisms. So essentially the type 4 hypersensitivity is mediated by chemokines and cytokines secreted by the Th1 type of cells. Most chronic diseases in humans which involve type 4 hypersensitivity are listed here. Most of these diseases are due to infections from mycobacteria, protozoa and fungi. Some have already been mentioned. This illustrative list consists of tuberculosis, leprosy, leishmaniasis, listeriosis, elastomycosis, which is actually a deep fungal infection, schistosomiasis, which we have already mentioned, and is due to infection from worms. Sarcoidosis is not completely understood. It is believed that presumably it's an infectious disease. These all diseases are caused as a result of chronic antigenic stimulation. Leprosy is of three types: tuberculoid, borderline, and lepromatous. Both tuberculoid and borderline types involve type 4 hypersensitivity. Both borderline leprosy and sarcoidosis show lesions typical of granulomatous hypersensitivity. Tuberculin type hypersensitivity, its clinical and histological symptoms are shown in this slide. This type of hypersensitivity was first observed by Koch himself. Koch found that subcutaneous administration of tuberculosis, a lipoprotein antigen from Mycobacterium tuberculosis, to TB patients led to the tissue at the site of injection. It became hard and swelling was also observed. This kind of a manifestation was considered a characteristic of the tuberculin reaction. The tuberculin reaction is now the basis of Mantoux test. In this test, a purified antigen, which is a protein derivative of tuberculin, is injected into skin, and consequences developed thereby are observed at the site of injection after 72 hours. The appearance of the red swelling is considered as an indication of a positive test. History reveals that these leukocytes and macrophages, the histology reveals that these leukocytes and macrophages infiltrate locally in the skin at the site of injection. Soluble antigens from many other organisms produce similar responses. The type of these organisms include M. leprae and Leishmania tropiaca. This hypersensitivity is also observed with some non-microbial antigens. Zirconium and beryllium reacted with skin proteins. Hence, tuberculin reaction is frequently used to test sensitivity to diverse antigens if there has been a previous exposure. Tuberculin injected into the skin is processed and presented by dendritic cells to T cells along with class II MHC proteins. The whole phenomenon is outlined here in this slide. These T cells are memory T cells which are specific to tuberculin. T cells produce cytokines which alter endothelial cell adhesion molecules. This allows monocytes to enter the local site and mature into macrophages. It is the synergy of T cells and macrophages which produce products which induce swelling. The time course for the different phases of tuberculin reaction is shown here. In about 12 hours after intradermal exposure to tuberculin, T cells migrate out of capillaries and accumulate around appendages like sweat glands. Collagen bundles of the dermis are broken. By 48 hours, CD4 positive cells outnumber CD8 positive cells by a ratio of 2:1. CD1 plus cells, which are actually Langerhans-like cells, also begins appearing between this period of 24 to 48 hours. In the final phase of 48 to 96 hours, keratinocytes get involved. The movement of cells between local sites and lymph nodes is very similar to contact dermatitis. In some cases, the tuberculin lesion may develop into a granuloma. This results from the persistence of some antigens or microorganisms which T cells are unable to destroy. CD4 plus T cells control many intracellular microbial infections. However, mycobacteria have escape mechanisms to prevent elimination. These consist of modulation of MHC class II synthesis, trafficking and expression, and inhibition of antigen processing for presentation by MHC class II protein. Hence, activation of macrophages by CD4 plus T cells is ineffective. This leads to chronic stimulation of CD4 T cells, which continues cytokine production. While immunological granuloma occur apart from due to M. tuberculosis and M. leprae, schistosoma infection, Crohn's disease, zirconium and beryllium exposure, and in sarcoidosis, non-immunological granuloma also occur with talc and a variety of particulate matter. In the latter case, macrophages are unable to degrade the inorganic particle. The granulomas are characterized by the absence of lymphocytes in the lesion. Granulomatous reaction sometimes can also occur from persistence of immune complexes and as in allergic alveolitis. The continuous production of cytokine leads to macrophages containing the microbe and fibroblast proliferation. Granuloma is a walled-off microbe along with the epithelioid cells and giant cells. Epithelioid cell is a large and flattened cell with more endoplasmic reticulum. These lack phagosomes. This slide shows the electron micrograph of an epithelioid cell. The accumulating macrophage secrete TNF, which results into differentiation of macrophage into epithelioid cells which fuse into giant cells due to continuous secretion of TNF. Experimentally, it has been shown that TNF is necessary for the development of granulomas. BCG injected mice, if injected with anti-TNF antibody, do not form granulomas. Giant cells are multinucleate cells, which are multinuclear cells, which are sometimes called Langerhans giant cells. These have little endoplasmic reticulum and degenerated mitochondria and lysosomes. These perhaps are terminally differentiated cells of monocyte macrophage line. An immunological granuloma has epithelioid cells and macrophages at core, sometimes along with giant cells. In some diseases like tuberculosis, this area shows necrosis. Core is surrounded by lymphocytes along with fibrosis. Mitsuda reaction is a granulomatous reaction in case of leprosy, and Quim test is the similar reaction in case of sarcoidosis. It was mentioned earlier that original classification of Gale and Coombs mentioned 4 types of hypersensitivity. Type 5 hypersensitivity is in fact a variation of type 2. In this case, the antibody mediating it is again self-antigen. Hence, many books still divide hypersensitivity into 4 types and cover the diseases arising out of this type 5 hypersensitivity under autoimmune diseases. We will discuss the frequently cited examples of type 5 hypersensitivity here and revisit the issues in more detail when we discuss autoimmune diseases. The classical example of the type 5 hypersensitivity is Graves' disease, where antibodies to the thyroid stimulating hormone receptor cause overactivity of the thyroid. The result is hyperthyroidism. Enlarged thyroid gland is visible as the swelling at the base of the neck and protruding eyeballs are typical appearance of a patient suffering from Graves' disease. TSH production is feedback inhibited by thyroid hormones. In the autoimmune disease like this also acts as a trigger for the thyroid cells to produce thyroid hormones. As in normal stimulation, the hormones feedback regulator regulate or stop the production of TSH by the pituitary. However, the autoantibodies to TSH continue stimulating thyroid cells to continue production of thyroid hormones. This is also the reason why the Graves' disease is an example of chronic hypersensitivity with chronic stimulation as the key mechanisms. Graves' disease is also called autoimmune thyroiditis. In humans, two major forms of autoimmune thyroiditis are known. Apart from Graves' disease, there is Hashimoto thyroiditis. In Hashimoto thyroiditis, autoantibodies activate complement and destroy thyroid cells. TC cells are also believed to be involved. The two are distinguished by referring to Graves' disease as thyrotoxicosis and the other one as Hashimoto thyroiditis. In Hashimoto form, autoantibodies are not formed against any receptor. The autoantibodies are directed against tissue-specific antigen as thyroid peroxidase and thyroglobulin. Women with thyrotoxicosis may give birth to babies afflicted with disease due to the antibodies against TSH receptors are passed through the placenta. Cases are known when people have both disease and said to have developed hashitoxicosis. The realization that Graves' disease is an autoimmune disease came from studies by physiologists who were interested in understanding how pituitary controls thyroid activity. These workers detected in the blood of the patients of Graves' disease a substance which was named LETS, which stands for L for long, A for acting, T for thyroid, S for stimulator. LETS, long acting thyroid stimulator. This was later on believed to be similar to IgG directed against the receptors on the mouse thyroids that mediate TSH action. However, there were patients with Graves' disease which had no LETS in their serum. So it turned out that there are two kinds of autoantibodies. One kind called LETS stimulates receptors of TSH in both mouse and humans. Another kind is inert against mouse thyroids and it is called LETS-PR, conveying LETS protector. Thus, experiments with mice have played an important role in our understanding of Graves' disease. Some other important results came from experiments with chickens. We'll briefly look at them when we discuss autoimmune diseases. Both diseases, which are called LETS and LETS-PR, account for 90% of the case of the Graves' disease. Now, let's summarize what we have learned in this lecture. To start with, we learnt about type 4 hypersensitivity and its three subclasses. First class, contact dermatitis, tuberculin reaction and the Mantoux test. Then the third kind, granulomatous hypersensitivity reaction. In this lecture, we also learnt about the type 5 hypersensitivity and we illustrated this with an important example of Graves' disease. With this module, we have now completed our discussion of all types of hypersensitivity reactions. There has been a common feature, and that has been the overriding of the protecting role of immunity. The immune system and its responses, as we have been discussing in the earlier modules, try to protect us from invading pathogens and other antigenic substances, the foreign substances. However, as we have seen in these modules concerning hypersensitivity, this immune system sometimes overdoes the thing, and that's a sort of a overriding of the protecting role of the immunity. So the immune system not working with within the range of vigorousness where it has a protective role can create problem. Tolerance, which you talked earlier, hypersensitivity, which we have talked in the last few modules, autoimmunity, and immune deficiency are all different facets of the same immune system, are also different facets of the same immune system which protects us. We saw, however, discussion of type 5 hypersensitivity reactions led to also talking about some autoimmune diseases. We, in the next module, now discuss these autoimmune diseases in some detail. Thank you.